Genetic yield potential is the maximum yield a plant can produce under optimal growing conditions. Actual yield is what ends up in the bin at harvest. Closing the gap between actual yield and a plant's genetic potential begins with protecting crops from the yield-robbing threats of stress both below and above ground. Carefully planned management practices that consider all aspects of crop production give crops the best path to succeed. Typically, conventional crop management decisions focus on two broad categories to drive production success. First, the physical components of production such as equipment, tillage practices, irrigation, and seed. And second, the chemical components such as fertilizer, herbicides, insecticides, and fungicides. Though the physical and chemical components of production are important, and the ag industry has experienced huge gains in production because of the focus in these two areas, the science community is now realizing that a third component, the biological component, is crucial to leveraging more production efficiencies. They are finding that a biologically active soil will see greater efficiency out of the physical and chemical components when compared to a biologically deficient soil. In other words, microbiology in the soil drives the physical and chemical components to higher productivity. Today, we can see clearly that when crop management focuses solely on driving the physical and chemical aspects of production while ignoring the highly important biological component, our soils become biologically deficient, limiting the plant's genetic yield potential. Managing the biological aspect of crop production may be new to some growers, but there is mounting scientific evidence that supports the value of such practices, as well as the risks that can be suffered when it is ignored. Let's take a closer look at what science is finding. A healthy, biologically active soil includes a balanced population of soil organisms, including bacteria, fungi, protozoa, nematodes, arthropods, and earthworms. For example, 1 teaspoon of healthy soil may contain up to 1 billion bacteria with over 6,000 individual bacterial species. This does not even begin to address the number of fungi, protozoa, arthropods, or nematodes that can be found in that same teaspoon of soil. Noted microbiologist Dr. Elaine Ingham states that these soil organisms support plant health as they decompose organic matter, cycle nutrients, enhance soil structure, and control the populations of other soil organisms, including crop pests. Therefore, scientifically speaking, management decisions that increase biological activity in the soil will have a direct impact on soil and plant health. Understanding the importance of microbes in crop production begins with the understanding of an essential, naturally occurring phenomenon known as root and leaf exudation. Exudation occurs when substances are released from the plant's root or leaf system. Plants naturally feed microbes through root exudates located in the rhizosphere, the soil area within 1/4 inch of the root, as well as through leaf exudates on the leaf surface. Once a green plant begins to photosynthesize, creating its own food, it produces simple sugars, amino acids, and organic acids called photosynthate. It will allocate a portion of this photosynthate to root exudation and deposit it outside of the root into the rhizosphere. This root-exuded photosynthate is a superfood for microbes. In the presence of this food source, native microbes in the bulk soil are attracted to the root's rhizosphere and begin to dramatically increase in number and activity. This increase in beneficial native microbes, stimulated by root exudation, triggers numerous benefits back to the crop, which include increased nutrient mineralization and availability in the soil, improved nutrient uptake by the plant, improved soil structure and moisture retention, improved root growth and development, increased production of natural plant growth hormones, improved overall plant health, growth, vigor, and quality, as well as an increase in the plant's natural defense against fungal disease, soil-borne pathogens, insects, and environmental stress like salt, pH imbalance, heat, drought, and saturated soils. As soils become more biologically active, applied fertilizers become readily available to the plant before leaching, volatilization, or tie-up occurs. Residue breaks down much faster, converting into organic matter. The physical characteristics of soil, such as compaction, change, and more aeration takes place. These are just a few areas directly affected by the microbial activity in the soil. As previously stated, through root and leaf exudation, plants feed microbes simple sugars, amino acids, and organic acids. In return, microbes provide the plant with essential plant-available nutrients, growth regulators, antibiotics, enzymes, and hormones, as well as break down applied fertilizer and release tied-up nutrients already in the soil. All of these components drive plant health and growth, as well as control pathogens. This mutually beneficial relationship is known as the symbiotic cycle. Though this symbiotic relationship naturally occurs between plants and microbes, management practices and climate factors can create unwanted stress and suppress the efficiency of this relationship, disrupting the symbiotic cycle. For example, tillage practices resulting in compaction, the use of harsh chemicals, fumigants, and high-salt fertilizers, as well as environmental conditions like pH and nutrient imbalances, waterlogged soils, drought, and excessive heat or cold are all stress factors that prevent the plant from producing high levels of exudates, thereby robbing yield potential. In a controlled environment, apart from stress, plants are able to produce enough exudates to increase the quantity of microorganisms in the rhizosphere up to 1,000 times greater than the bulk soil around the rhizosphere. Through photosynthesis, a plant creates the energy it needs to function. The plant allocates this energy systematically in a purposeful way. This chart illustrates how that photosynthetic output is disseminated. To the left of the chart is a priority scale that ranges from high to low. In simple terms, plants first use energy to deal with any type of stress. Only after stress is managed will the plant allocate its energy to mineral conversion and acquisition, grain fill, then to plant architecture, root growth, and exudates. Last in the process is achieving genetic yield potential. It is estimated that green plants can allocate as much as 20 to 40% of their total photosynthetic output to root exudation to feed microorganisms. Therefore, the importance of caring for the soil's microbiology is of utmost importance if genetic yield potential is to be achieved. Our soils have been pushed and overworked the past several decades in efforts to close the gap between actual yield and genetic yield potential. Nature is telling us that the physical and chemical components alone of conventional agriculture are not sufficient to overcome the harm caused when the biological component is neglected. Now that we better understand its value, will growers stop the use of harmful chemicals and tillage practices that have a negative effect on soil biology? Probably not. But that's where AgriGro's prebiotic technology comes in. Through innovative products like SeedMax, Ignite S2, FoliarBlend, Ignite S4 Technical Grade, and Ultra, growers can offset the negative effects of today's growing practices and support their soil and plant health by vastly increasing the beneficial microbial populations already present in their production environment, even greater than what nature can do on its own.